Hello everyone, I am Yashoda Wanigasekara. I am a master's student majoring in biomedical engineering at Yangon University, China. The study I am going to present you today is a paper written with my graduation research. The presentation summarizes a study on polystyrene nanoplastic and their effects on PC12 cells, supported by meta-analytic evidence on oxidative stress and neuroinflammation. This study incorporates an acute in vitro PC12 experiment with a synthesis of published neuronal studies. These slides introduce the biological foundation of this research. Plastic remains because of their durability and slow breakdown. Once particles reach nanoscale dimensions, they became more biologically active because of surface area, diffusibility, and contact with the cellular structures. The nervous system is sensitive target because neurons depend strongly on mitochondrial respiration, redox regulation, and membrane stability. In the picture, you can see the NPS sources, polymer types, and exposed pathways for humans. Our study addresses a practical gap. Present papers are not fully comparable because they differ in cell models, particle size, doses, duration, and endpoints. So, this study combines an experimentally controlled PC12 cell exposure model with the meta-analysis of published neuronal studies. The aim is not only to describe cytotoxicity, but to connect viability and cell death outcomes with broader oxidative stress and inflammatory evidence. Let's move into the experimental methods. The experimental design in our study used PC12 cells and three nominal particle size of 100, 300, and 500 nanometers. Cells were exposed using 10, 20, and 30 microliter addition of a 2.5% or 25 milligram per milliliter stock suspension. Because MTT and FDAPI assays use different well formats and medium volumes, the final nominal concentration differ between the assays. In the meta-analysis design, the literature followed PRISMA 2020 guidance. The review identified 2,646 records from three main databases and 35 records from other sources. After duplicate removal, screening, trial, and eligibility assessment, 25 studies with 1,426 observations were included in this study. The meta-analysis used HG and random effect models because of expected heterogeneity in models, particle features, exposure setting, and biomarkers. The risk of bias assessment indicate that effect size should be taken cautiously. Now let me present you the result of this study. The MTT assay showed a clear reduction in cell viability after 6 hours exposure, especially for smaller particles. As you can see in the graphs, the 100 nanometer group showed strongest viability. The 300 nanometer group showed intermediate toxicity, while the 500 nanometer group showed weaker effects, especially at higher nominal concentration. For the time course, results showed viability decline from 1 hour to 6 hours. FDAPI staining supported the MTT finding by showing increased membrane compromised cells. In this 100 nanometer group, cell death increased with the increase of nominal concentration. But at the same nominal concentrations, the 500 nanometer group showed much lower death rates, around 10 to 12 percentage. The time course also showed progressive increased cell death from 1 hour to 6 hours for the 100 nanometer condition. For oxidative stress, the pole effect was weak and less stable at dose of 25 microgram per milliliter of all over. Effects were positive at 25 to 50, 50 to 100, 100 to 500 dose groups. By particle size, the strongest pole effect was observed in the 0 to 50 nanometer subgroup. By duration, the largest point estimate appeared at 12 hours or less and remained positive all over long exposure windows. The neuroinflammation meta-analysis showed positive pool effect across the all major dose categories. The strongest dose point estimate was 50 to 100 dose group. By size, the 100 to 1000 nanometer subgroup had the largest pooled estimate. However, particles greater than 1000 nanometer showed a weak and non-significant estimate. By duration, effects were positive and became pronounced in the longest exposure subgroup, 48 hours to 30 days. The combined analysis should be taken cautiously. The PC12 experiment directly proves reduced NTT viability and increased FDA PI evidence of membrane damage and cell death, mainly for the 100 nanometer group. The meta-analysis support the broader biological probability that PS-NP exposure is associated with oxidative stress and neuroinflammation in neuronal models. However, ROS, mitochondrial markers, cytokines, and pathway-specific cell death markers were not measured directly in the PC12 experiment. So these mechanisms should be treated as supported by the literature rather than experimentally proven here. In this study, the main limitation is that the PC12 experiment is an acute monoculture system. It is useful for comparative toxicity, but cannot reproduce the cellular heterogeneity and barrier function of the nervous system. The dosing groups used in the study were nominal and stock volume based. Also, particle behavior such as aggregation and sedimentation was not measured. The meta-analysis strengthens the overall understanding, but heterogeneity, risk of bias, and publication bias signal means that exact effect size should be taken cautiously. Future works should measure direct mechanics under matched condition and use more physiologically relevant neuronal models. Experimentally, PS-NPs reduce PC12 cell viability and increase cell death associated signals for FDA-PI. The evidence was strongest for smaller particles, higher concentrations, and longer exposure. The meta-analysis placed these findings into a broader neuronal toxicity framework connecting oxidative stress and neuroinflammation. The final message is that PS-NP neurotoxicity is supported, but the exact mechanism requires direct validation with oxidative, mitochondrial, inflammatory, and cell death biomarkers under matched conditions. So, this is all for my presentation. Thank you for your attention. Thank you.